So we have a little bit to finish up from yesterday, nothing too crazy. And then we will move on to day 3, which is signal transduction. Relatively short. We didn't need to get into the super nitty gritty details. So hopefully, we'll be able to knock this out and be able to have extra time for assorted exit tickety things. Let's pull my mask off with the mic. Okay. So before I get started, how are we doing? Anybody? Anything? Questions? Comments? Concerns? All right. So we had just finished up receptor tyrosine kinases. Yes. Yes. All right. So those were 2 of our 3 membrane cell receptors. So we have one more type of membrane receptor. I'll talk a little bit about receptors that exist just like free floating in the cytosol, and then we will move on to the next phase in cell signaling. So our third cell membrane receptor are ligand gated ion channels. Ligand gated ion channels. Ions will often act as cell signals or are heavily involved in cell signaling, which we will talk about today. But this kind of receptor acts as a gate, opening or closing as shape changes. So receptor act as a gate. Opening. My board's kind of off. I might have to fix that. And closing as shape changes. So the doodle's not necessarily as complicated as last time. Say we've got our cell membrane. This is outside of the cell. This is inside of the cell, just like our orientation was yesterday. Change color. Make it purple. If this is our receptor. In this form, it is inactive. Outside of our cell, we will have a bunch of ions. I'll say these little green dots are ions. Ions can't diffuse through the cell membrane. They have to be let in through specific transport stuff. And then we have, just keep changing colors, our ligand. But it's not bound yet, so everything is inactive. This is just kind of our set the stage kind of thing. So our receptor exists as kind of a gate. It's kind of difficult to see how it is a 3D shape, but we could also draw like a little line through here if we wanted to. That's where it'll open up via channel. So our next phase, we will have right? Our ligand will bind to our receptor our receptor will open our gate. Now that our gate is open, our ions can flow through to the inside of our cell. So, like a key in a lock, ligand fits into the receptor. Receptor changes shape, opens up, and allows ions in. Those ions will then trigger a cellular response. And then, as quickly as it started, as soon as that receptor or the ligand were to leave, our ligand-gated ion channel will close back up and become inactive again. Ooh, that is hellacious. All right. Nothing super complicated for today. It's definitely not G protein coupled receptors, but it is another very, very important type of receptor that happens on cell membranes. Yes. Yep, this is active. Yep, when the ligand is bound to our receptor, that is typically what activates it. Opens up and lets stuff in. Yes. Yes. Good question. Any other questions? Okay. So those were our membrane bound receptors. Now we are going to briefly talk about intracellular receptors. So cell membranes aren't the only place in cells that receptors exist. These membrane receptors can also exist on other cell membranes, so nuclear membranes, all that fun stuff. But there are receptors that are just free floating within the cell. I'm going to do this because I'm going crazy. Okay, hopefully that'll help. So, intracellular receptors found in the cytoplasm or in the nucleus. So there are receptor proteins just kind of floating around the nucleus with the DNA. They exist mostly to trigger things like DNA synthesis or mRNA synthesis. So intracellular receptors are found in the cytoplasm or the nucleus because signaling molecules can pass through the membranes. They don't have to be outside of the cell because the signaling molecules can just come in whenever they want to. Signaling molecules can pass through membranes. Why do the extra work of binding on the outside and then transmitting the signal through the cell if the signal can just come into the cell on its own? So these signaling molecules are usually small or hydrophobic. because those types of molecules can pass through the cell membrane without having to be actively transported in. Hormone signaling is a big portion of this. And intracellular receptors usually lead to turning on or off particular genes. Often lead to turning on or off particular genes. So while they are not as common as our membrane receptors, they do carry important functions through the cell, being able to turn genes on and off. Let's see. Hormone signaling is a really big deal because, like, I keep using puberty as an example because it's one of the easiest. Hormones are released; they don't have to be bound on the outside of the cell; they just fade in. They can pass through the nuclear envelope, hit those receptors inside the nucleus. The nucleus is like, okay, time to start transcribing this DNA. Proteins are made. Puberty happens. That kind of stuff. Any questions here? Nothing too crazy. Didn't have a heck of a lot left over from yesterday. All right. So that was cell signaling day 2 with reception. Now we are talking about transduction. So I'm just going to start a whole new page. Transduction is the second stage in cell signaling. Reception, transduction, response. Not a heck of a lot of doodles, not a heck of a lot of paragraphs, but a lot of vocab words. We're not doing super in detailed stuff, but I want to make sure you get the gist. So, transduction oftentimes is just a cascade of molecular interactions that relay signals from receptors to their target molecules, the middleman, if you will. So, the process of transduction cascades of molecular interactions relay signals. Oh, my handwriting's so bad today. From receptors to target molecules. So, if the light switch is a receptor and the light bulb is our target cell, transduction would be like the wires that carry the electricity from the light switch to the light bulb. So they just help the signal move from one place to another. And often, more often than not, this happens, or almost exclusively, I guess, happens through signal transduction pathways. We talked very briefly about signal transduction pathways on day 1. And we might repeat some stuff, but I think it's for the best. All right, so in signal transduction pathways, protein-to-protein interactions are a major theme of cell signaling. We got a lot of proteins. We use a lot of proteins. So, protein-to-protein interactions are our major theme of cell signaling. And a main way that those proteins interact is through protein phosphorylation and dephosphorylation. Now, phosphorylation and dephosphorylation is a fancy way of saying we're adding and removing phosphate groups from these proteins. And a major player in protein phosphorylation are protein kinases. We talked about kinases yesterday when we talked about receptor tyrosine kinases. Tyrosine kinases are kinases that phosphorylate tyrosine. Protein kinases are enzymes that transfer phosphate groups from ATP to a protein. So kinases are a class of enzyme. Protein kinases are enzymes that transfer phosphate groups from ATP to proteins. I think we might even have this exact definition written down again. That's all right. So, protein kinases are enzymes that transfer phosphate groups from ATP to proteins. So, tyrosine kinases are kinases that add phosphate groups to tyrosine proteins. So, protein kinases, just a class of enzymes. Protein kinases help drive something called a phosphorylation cascade. If you need a fun biology themed band name, phosphorylation cascade is my personal recommendation. So, in a phosphorylation cascade, a series of proteins in a pathway are phosphorylated, each adding a phosphate group to a next. So, protein gets a phosphate group, which then adds a phosphate group to another protein, which then adds a phosphate group to another protein, which then adds a phosphate group to another protein until we get where we need to go. Like a messed up game of hot potato. So, a series of different proteins in a pathway are phosphorylated. Each adding a phosphate group to the next. So one protein kinase will activate another protein kinase, which will then activate another protein kinase, and so on, and so on, and so on, and so on. But like everything in biology, what can be done often can be undone. So just like we have to turn these signals on, we also have to turn these signals off so we're not wasting time, energy, and resources. So we have special molecules that will undo what our protein kinases do, and they are called protein phosphatases. ACEs, meaning enzyme, phospho, so it is breaking down or taking phosphate groups off of enzymes. Or it is an enzyme that takes phosphate groups off proteins. Protein phosphatases are enzymes that rapidly remove phosphate groups from proteins. This, of course, is called dephosphorylation. So what is done must be undone. So our protein kinases and protein phosphatases are our key players in a phosphorylation cascade. So we're going to draw one. Very, very simply, nothing too crazy. Phosphorylation. Are we good? Are we good to? Okay, so we've got a phosphorylation cascade. Starting off, of course, with a cell membrane. This is outside the cell. This is inside the cell. Try to stick with a similar color scheme. We have a receptor. This could be any type of our receptors. This could be, this technically could be an internal receptor as well. It could be a G protein coupled receptor. It could be a receptor tyrosine kinase. It could be an ion gated receptor. So we're just going to generalize it. So this receptor is activated by a signaling molecule. So signaling molecule activates our receptor in any of the processes that we talked about yesterday. This will activate a relay molecule. So all those molecules that we drew yesterday that I said cellular response, this is that molecule. So over here in this big circle, we're going to call this protein kinase 1. I'm going to use my little protein symbol. Protein kinase 1. And this is inactive. But what that relay molecule is going to do is activate it. So signaling molecule activates a receptor. Receptor activates a relay molecule. And that relay molecule activates protein kinase 1. Like I said, this is just a general structure. This can happen in a lot of different ways with a lot of different proteins. There is no protein kinase 1. It's just a protein kinase. So then, protein kinase 1, we've got a protein kinase 2, inactive. But protein kinase 1 is a protein kinase, so it will activate protein kinase 2. Because what do protein kinases do? Add a phosphate group. This could go on. There could be a protein kinase 3. There could be a protein kinase 4. I think y'all get the gist. Protein kinase 2, we'll say, is the end of it, which will activate a protein and cause a cellular response. Now, when protein kinases do work, so when protein kinase 1 activated protein kinase 2, it took an ATP. Because that phosphate group is now on protein kinase 2. Because kinases take a phosphate group from ATP and add it to a protein. But if this protein kinase 2 is activated, it will continue to trigger this cellular response, but we don't need cellular responses all the time. It's a response for a reason. So we have to be able to shut this protein kinase 2 off. And we do that with a protein phosphatase. So we'll make this one, make it this yellow color. So we have this protein phosphatase. Protein kinase 2, we will lose a phosphate group and become inactive again. So, I don't know how much I can get into. Aha. There we go. Our signaling molecule activates our receptor, which activates a relay molecule. That relay molecule will activate a protein kinase. That protein kinase will use ATP to activate a second kinase. That second kinase can use more ATP to activate a third, but in our case, it's going to activate our cellular response. And then our protein phosphatase is going to inactivate protein kinase 2 in order to stop the cellular response. How are we doing? Okay. So that is your basic outline signal transduction pathway. They could be longer, they can be shorter, they could be simpler, they could be more complicated. But that is our general outline. I'm not going to make you memorize specific signal transduction pathways. There's hundreds of them. But if you want examples, I'm more welcome to find them and figure it out. Ooh, that's not what I want. Aha. Okay. So there are other molecules involved in cell signaling, and they are called second messengers. Am I still yellow? Yes. So, small molecules as second messengers. Not all components of signal transduction pathways are proteins. Proteins come in handy. Proteins are a very big player, but there are also second messengers. So protein in this scenario, your proteins are sorry. I'll go to it. So the definition of a second messenger. A second messenger is any small, non-protein, water-soluble molecule that is involved in a signal transduction pathway. Small, non-protein, water-soluble molecule or ion. Second messengers play like critical roles in these signal transduction pathways. They are called second messengers because our primary messenger is the ligand or the receptor. So the ligand that binds to that receptor, wherever that receptor may be, is our primary messenger. So our second messengers are something that happens down the line. They readily spread through the cell via diffusion. They are small and water soluble for a reason. And they can participate in pathways triggered by both G protein coupled receptors and receptor tyrosine kinases. So can participate in pathways initiated by G protein coupled receptors and receptor tyrosine kinases. Now there are a lot of second messengers, but the most widely used and the ones we're going to talk about the most widely used are cyclic AMP or little c camp. That's a weird looking M. Close my parentheses. I don't like that at all. That's better. Cyclic AMP and calcium ions. Not only is calcium important for strong, healthy bones. But it is important because it is a critical second messenger for a lot of signal transduction pathways. So these are the two we're going to talk about relatively quickly. You don't need to know like the ins and outs, but I wanted to give you a generalized background on a lot of them. My freshman bio class in college, I think we spent like 2 weeks talking about cyclic AMP. So I want you to be familiar with it at least. So, camp. Also known as cyclic adenosine monophosphate. So, ATP, take another phosphate group off of it, and a ring forms cyclic AMP. It is an yes. So it is formed by an enzyme embedded in the plasma membrane. So enzyme in plasma membrane. Will convert ATP to cyclic AMP in response to an extracellular signal. So external or membrane receptor ligand binds. That ligand will activate our enzyme. That enzyme will very, very rapidly convert ATP into cyclic AMP. We can increase our concentration 20-fold in seconds. I'm going to use concentration brackets. And that will broadcast the signal through the cytoplasm. Go away, please. So that's typically how cyclic AMP is used for stuff that has to happen really, really quickly, and that signal can be broadcasted through the cell like that. So there's that one. I think I'm gonna have enough time to get through calcium ions. So before I do that, any questions about camp? Okay, calcium ions. So, these are really important neurotransmitters. Calcium is good for your brain, good for your muscles, because it is our signaling molecule to get through neurosynapses. It can be used as a growth factor. It can be found in hormone pathways. This stuff is more widely used than cyclic AMP. Our calcium ions are super, super important. In fact, an increase in the concentration of calcium ions triggers a variety of signaling pathways. Increase in the concentration of calcium ions triggers a variety of signaling pathways. Okay, so usually cytoplasmic concentration is super low. In fact, it is actively transported out, or actively transported into the ER. That's where we store it. We need it for signaling molecules, but we need to keep the concentration in our cytoplasm low, because if not, it would be hard to detect concentration changes. So if we keep it super low, any calcium ion can be easily detected and we can notice the concentration change. So, small change is a big deal. So, in response to a signal, I say we, or a cell, can release calcium ions from the endoplasmic reticulum. So, just like our signal receptors can trigger the enzyme that converts ATP to CAMP, a signaling molecule can trigger the release of calcium ions, which can do a myriad of things. That's all I have for you for transduction. I try not to give homework, but if you could knock out those exit tickets for yesterday and today, tonight, they're like 5 points each, like 3 questions, nothing too crazy, just so we are ready for response tomorrow. If you have any questions, please let me know. I know, this week is kind of notes heavy, but I will try to make next week a little less crazy.